My name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this session, a lot of things are going to come to fruition, or at least I certainly hope they will. We're finally to the point where we're going to see at the molecular level how translation occurs mechanistically. We've already got the concept down, and that's why I put the central dogma here, because we know that DNA information gets copied into RNA information, which we've already covered the mechanism of that. Then we've talked about the idea that RNA information is going to be copied into protein information. We've even discussed the code, the way that it must sort of happen. But we did not discuss the specific way the machinery operates, and that's what we're going to talk about today. And I really believe, I know personally, when I understood this, I felt it to be very, very powerful, and a lot of things really come together at this point, from original Mendel type experiments to everything basically. So we're going to look at the mechanism of translation. This is all part of the larger idea of how genotype turns into phenotype, if you want to think of it that way. We know that the expression of the information in a gene leads to a protein, and now we're going to see how that happens. We're going to use bacteria as an illustration for a couple of reasons. I've probably said it several times already, but all of this was actually discovered using E. coli bacteria. We know a lot about it in bacteria. We've subsequently learned quite a lot about it in other eukaryotes. So we'll do it the bacterial way, and then towards the end of this session, I'll talk to you about some general differences in eukaryotes. Okay? I don't want to keep you waiting because you've been waiting for this a long time. Now, in a certain sense, but don't make it too much of a sense in your mind. In a certain sense, the idea is similar to transcription, but only, in the following way. With transcription, we had a promoter which said start here. We had elongation of the messenger RNA, or whichever RNA. And then we had a terminator that said stop here, right? Well, it only makes sense. In protein synthesis, we're also going to have a start, an elongation, a continuing, and a stop, right? So, but the actual machinery is quite different for these things, all right? So remember what happened so far. A messenger RNA that's ready to be translated is going to be ready to go. It's going to be interacting with the ribosome. We don't have to go, we already just went over how that messenger RNA came into being. So let's look at the initiation step. How does it begin this process of translation? So first thing that happens, the messenger RNA binds to the small subunit of the ribosome, and this is a good point. A lot of people have the idea that the ribosome is sort of just pre-made with the big subunit and the small subunit, but this is not the case. The two subunits are not affiliated with one another until initiation occurs. Now, this has to be, just like transcription, we said it has to start right at number one. We have to start at the exact right place here. So the way that gets done in bacteria is there is a complementary base pairing between the 16S ribosomal RNA and a sequence that is upstream of the AUG. Remember, AUG means start codon. And it's often referred to according to the names of its discoverers, the Shine-Dalgarno sequence. Often it's also just referred to as the ribosome binding site. But the point is, it's a part of the messenger RNA that is complementary to the ribosome. So when they base pair, it positions the AUG start codon very perfectly right in the ribosome, exactly where it needs to be. All right? So you can think the messenger RNA just, and I'll show you a picture in a moment, just came over and bound in the correct way to the small subunit. And then a tRNA, a transfer RNA, which has formylmethionine. I'm pretty sure we mentioned this, that so-called FMET, it's just a modified methionine in bacteria. That's the methionine that comes over. And an anticodon on that transfer RNA has to be like this. 3' UAC 5'. And the reason is, I think you get it, but if the start codon is AUG this way on the messenger RNA, right, that tRNA is going to come over, and we're going to need it to be complementary, so it's going to have to be UAC, but the polarities are the opposite way. So because that messenger RNA codon of AUG is there, always a transfer RNA with this anticodon is going to come over. In fact, that's why it comes over, because of the attraction, if you will, of the complementary base pairing. So now when we've got the messenger RNA, the small subunit, and then the tRNA all together, that's when the large ribosomal subunit joins the complex. And now we are ready for translation. Now, the ribosome is a fairly complex macromolecular structure. It's got about 43, I say about because there are definitely 3 big RNA pieces. There are about 40 proteins. I really don't remember exactly how many. But it has 2 functional sites, sometimes 3, but we're going to focus on the so-called P site. This stands for peptidyl, but we'll talk about why a little bit later on. Now, in the P site, that FMET tRNA that just came over is there. And then there's an A site. That stands for aminoacyl. It may not be obvious right away why this is the case, but we'll get to the bottom of it. Right now, it's empty. There's nothing in there. If your textbook or your professor speaks of an E site, the E site is something that does exist, but I think it complicates the analysis, so I'm not going to speak of the E site right now. It's really not necessary to understanding the process. Now, you might imagine this gets rather technical potentially, so I'm not going to speak about it a lot, but there are proteins called initiation factors, or IFs, that are necessary to facilitate these steps. Think of them as catalysts. They're not technically enzymes, but they're needed for this stable so-called initiation complex to form. All right? So let's take a pictorial look at what's going on here. They say a picture's worth 1,000 words at least. So if you want, we can just read this little description. It should be what we just said. The messenger RNA binds to a small ribosomal subunit near the initiation site, right? So it's not labeled here, but somewhere up here is the Shine-Dalgarno site. See, when we make diagrams of this, it has to be a little bit off scale so that we can get the idea. And the reason I just realized that is if it's up here, it's not binding to anything, right? But we're making the bases look a lot bigger than they really are just so we can get the idea. So my point is the Shine-Dalgarno sequence is somewhere upstream of the AUG start codon. The messenger RNA has bound to the small subunit so that the AUG start codon is in a very definite spot. Then a transfer RNA, again, just because AUG is there, a transfer RNA that has UAC as its anticodon is going to come over, and that is going to be carrying methionine, in this case, formal methionine, with it. So this is the beginning of the initiation complex. And this first tRNA is often referred to, as you might expect, as the initiator tRNA. So we have those together, and then we said the large subunit comes on over to form a functional ribosome. The ribosome is often referred to as the workbench on which a protein is made, and I think it's a very apropos sort of an idea. So once the large subunit comes over, a functional ribosome is formed. It's important for us to note that the A site and the P site, we're going to keep track of them as we go along here. So they're labeled here. This is the P site, and we said that the P site is the place where the first RNA that's come over exists, and that the A site is now empty. But you may notice, presupposing what's going to happen next, that now also, guys, I think you'll understand this. Obviously, there isn't a big space between one codon. Maybe it's not obvious, but there's not a big space between one codon and the next. They go one after another. But to get the concept across, we're making it look like this is in the P site, and what I wanted to bring up was in the A site right now is another codon that can be read. It's GAA. Okay, so that portends what's going to happen next, and I'll spill the beans a little bit. If that says GAA, probably a tRNA that has the appropriate complementary anticodon is going to come over there, right? But maybe we're getting ahead of ourselves. So we have this really is initiation complex. It's ready to go. So we've got the starting down. Now you've got to get the continuing down. And the continuing, in this case, don't forget what we're doing. We're building a protein here, right? The building of a protein is going to require that amino acids become covalently joined to one another. So let's talk about how that can happen. With respect to elongation, so we just saw that there's another codon positioned there in the A site, right? And a tRNA corresponding to that next codon arrives at the A site and binds. What do we mean corresponding to it? It has to have an anticodon that's complementary. And that's, again, why it's coming over. Now, the amino acid that's on the tRNA in the P site is now transferred to the amino acid on the tRNA that's in the A site. And that forms the first peptide bond here. The word, it can get wordy, but when we look at the picture, we'll make sure we understand it. But this is called a step that's peptidyl transferase, and it's catalyzed by an enzyme called peptidyl transferase. Interestingly, it doesn't really have to do with the mechanism. It's just fascinating. It's been found that the reaction is actually catalyzed. The enzyme is part of the 23S ribosomal RNA, which means that this is a ribozyme. I think we've discussed that briefly, but there are RNA enzymes, and apparently the ribosome, the peptidyl transferase activity, is an RNA enzyme activity. But one way or the other, the point is that the next tRNA comes, we're going to shift the amino acid from the tRNA in the P site to the one on the A site, and then, very importantly, translocation is going to occur. The ribosome is going to move so that what was in the A site is now in the P site. The tRNA that was in the P site gets kicked out at this point. Just as I said before, it could get very technical, but proteins called elongation factors are necessary to facilitate these steps. But we really want to make sure this is the crux, understanding this part of it. Just reading this off, even in my head, I'm thinking it might not come across exactly, oh, I understand now, because we really have to think about it, and pictures really help. So we saw it before that we had the one tRNA there. It's not labeled here, but remember, this is the P site, this is the A site. We saw that the GAA codon was there in the A site. So now, as we predicted, a tRNA is going to come over here that has a complementary anticodon. So if GAA is there, we're not worrying about the polarities right now, but they are relevant. But what's complementary to GAA? CUU, right? So this tRNA with the appropriate anticodon is going to come over. That happens to be carrying the amino acid glutamate, and that tRNA is going to stably bind. So now we've got one tRNA in the P site, one tRNA in the A site. And what you should notice here is, so that was the first step that we just discussed, the second tRNA coming over. Now we're going to do the peptidyl transferase activity. It's hard to make this fluid, which is why I'm sort of doing a little bit on this picture, and then we'll go to another picture. But the bottom line, guys, is this, and I remember having a hard time with this when I was a student. We're going to break this bond that's holding the methionine onto the tRNA, and we're going to attach this methionine to this glutamate, all right? And if we redrew it, I mean, this is a dipeptide now, and it's attached to this tRNA here. It's no longer attached to its original one. So that's the peptidyl transferase part of this. Now, peptidyl transferase, these are the early steps, right? If there was a large peptide there, We'd be moving the whole thing over, as you'll see in a minute. But the two RNAs are now bound. So then we said, what's the next thing that has to happen is translocation. The ribosome shifting so that what was in the P site, what was in the A site is now in the P site, and what was in the P site is going to get out of here. So now you see, look, this dipeptide is now attached here. So this original initiator tRNA is getting out of the way. It did its job. What do we need with this thing anymore? It's done. When the ribosome moves, this whole thing is going to end up in the P site. And the A site will be empty again, which is what's shown over here. Look, so now you can see why this is called the P site. The growing little polypeptide, which is only 2 right now, but it's going to get a lot longer, is now present on the tRNA that's in the P site. All the time when the A site's empty, I hope you're getting the idea already. Now there's another codon there. And a tRNA with a complementary anticodon is going to come over with its appropriate amino acid. And the A site, because there's always a tRNA bringing one amino acid in there that's going to get added to the chain. Now in terms of protein chemistry, the methionine is always going to be at the N-terminus of the protein. Every time it gets translocated, because eventually this is going to be stuck onto there. It's growing so that the last amino acid that gets added will be at the end, the C-terminus, if you will. All right? So translocation occurred here. This tRNA moved from the A site over to the P site, and here it is in the P site, right? Carrying still now the dipeptide, the growing, the nascent polypeptide chain. And we're just going to repeat this process over and over and over again. I believe we have an animation about this, but you can find lots of animations just by looking on the internet that are very good. I would say after you think you pretty much understand it, or I hope it's not the case, but if you're having trouble understanding what I'm telling you, if a picture is worth 1,000 words, sometimes a video is worth 1,000,000. So, but get the idea. So this coming in here, right? This tRNA going into the A site, bringing its tyrosine over, and what's going to happen? So we're just going to keep doing it over and over. I said what's going to happen, and I didn't answer, but I think you sort of have the answer. It's going to come in there. We're going to break this bond and transfer this over to here. We'll have another picture in a minute. But the point is, this keeps going. Now, we can't go for 1,000 amino acids, which is about the size of an average protein. It could be longer or shorter. But it just literally over and over and over again, right? A new transfer RNA bringing an amino acid comes over. The peptide gets transferred, and the ribosome shifts over and over and over again until it's time to stop. So I just wrote this because I think it's so cool. I doubt you would really ever have to know this. But in E. coli, under conditions that are optimal, the rate of elongation is 20 amino acids a second. 20 amino acids. And the fidelity has to be perfect, you guys, right? So I don't know if I can do math. If you had let's say you had a protein that was 200 amino acids, right? I guess it would take 10 seconds for that to be made. That's pretty wild to me. It's really going fast by our terms. However, eventually a stop codon is reached. You may have them in your mind, you may not, but remember UGA, UAG. On the chart, there are 3 stop codons. When one of those is reached, the point is, guys, there is no transfer RNA that has an anticodon that's complementary to the stop codon. So you could think of it this way. When the stop codon gets into the A site, what's been happening all along? A new transfer RNA was bringing a new amino acid, right? What happens now? Nothing, because there's no tRNA that says I'm attracted to that sequence. So the ribosome sort of stalls, and eventually a release factor binds to the stop codon. And basically, think of it this way. What would normally happen? The whole peptide now would get transferred to the next amino acid. But there isn't any, so it gets released instead. So this causes termination and release not only of the new polypeptide, which was the point, but everything pretty much falls apart. You know, the large and the small subunit, the tRNAs go, and in essence, you're done. I mean, in terms of the goal of making this one protein. I mentioned this before, just that we're talking about the A and the P sites. Many textbooks will talk about the E or exit site. I really think, I mean, I'm not trying to downplay the E site. Well, actually, I am a little bit. It's important, but it's not important to understanding informationally and mechanistically how this occurs as far as I'm concerned. If your professor is obsessed with the E site, then I guess you have to know about it. Okay, so we're back to a picture here. We've gotten to the point where we've been building. Now, obviously, guys, 3 amino acids is not a real long protein, but we're just trying to show the idea. It would get a little out of hand if we had 1,000 amino acids over here. So the idea is we had if we go back to this picture here, right? This, draw it in red, there's a lot of yellow around. That tRNA came over. That peptide, that little tiny peptide, was transferred over, and the ribosome translocated, which is what you see here. So now, just as you would expect, now there's another codon in the next site. So pretend like 1,000 times this already happened. We've got this big polypeptide over there. And now, the stop codon has come into the A site. Now, what tRNA is going to come over? There isn't any. So a release factor binds to UAG, or whatever stop codon, and then just pretend this whole polypeptide is looking to be transferred to something, but there's nothing to be transferred to, so that bond gets broken and it gets released, and you can see here everything just falls apart, and that's fine. And you just made a protein. It's amazing, and I'm being serious. This is when this was understood. Like I said, so many things got brought together. I mean, it's really a beautiful, amazing thing. Now, there are, feel free. You could do whatever you want, but what I'm saying is this doesn't always click right away. So, of course, maybe you want to watch it again. Maybe you want to find some animations, look at animations. I'm sure that you'll get it. If you just think of it, it is a little machine. It just goes in a stepwise fashion, reading codons. This is the whole collinearity here, right? How the codon equals this amino acid, then the next codon equals this amino acid, and so on and so forth. So that's the idea, but there are still some questions that remain. There are always questions, right? Because I'm telling you that, okay, well, AUG, and you know from the genetic code table, AUG means methionine. So what I said is, well, the tRNA with the complementary anticodon always brings methionine. But it's a good question to say, well, how come the right amino acid, the correct one that makes the code work, how did it end up on the right tRNA, meaning the one with the correct anticodon? Because if this wasn't absolutely controlled, the code would be a disaster. It wouldn't work, right? You'd have a big mess on your hands. You wouldn't have any hands because you wouldn't exist. There are enzymes called, it's a long word, but it does explain what they do. They're called aminoacyl tRNA synthetases because they synthesize an aminoacyl tRNA. We didn't use that term, but a tRNA can exist with or without its amino acid. When the tRNA is by itself, it's just the tRNA. When the enzyme comes along, it catalyzes putting the appropriate amino acid onto that tRNA. And now we say that it's charged. It doesn't have anything to do with electrical charge. It just means it's got the correct amino acid on it, and it's able and ready to go into translation now. So these enzymes put the right amino acid on before the transfer RNA ever is really involved with translation. Now, I think you would guess these enzymes have to be highly specific. It's not 100% understood how they differentiate one tRNA from another, because remember, they all have basically the same shape, but the fidelity of the code depends on it. We do know a bit about it. Um, One thing is clear though. It makes sense to think of it this way, except we'll see one reason why it might not be a good idea in a little while. But it would make perfect sense to say that the amino acyl tRNA synthetase checks the anticodon, right? And says, well, for example, CCC means proline, right? If a tRNA has GGG anticodon, the enzyme could say, okay, GGG, that means you got to have proline on you. That would make sense, but not everything that makes sense means that it actually happens. Experiments have been done where the codon can be changed, and that's the only thing that changes, and the enzyme still recognizes that particular tRNA as what it should be and puts the appropriate amino acid on it anyway. So it can't be that the enzymes are recognizing the anticodon. At least it can't be the main thing that they're recognizing. So there's a lot of work that's been done to show different features of the structure of these individual tRNAs that the enzymes recognize. It's exquisitely specific, and it just shows you how specific enzymes can be in their interactions with their target, their substrates, right? Now, logically speaking, there have to be at least 20 different amino acyl tRNA synthetases, to at least one for each amino acid, right? But we've discussed this before. There are always more because, remember, you don't have to know the exact number. But even when we were talking about tRNAs, we said, look, there have to be at least 20 of them. The maximum would be 61, but the real number is somewhere in between that due to degeneracy and a variety of other factors. But however many tRNAs there are, put it this way, there have to be at least that many. There have to be that many amino acyl tRNA synthetases. So this all has to be done prior to the process that we just described. The amino acids coming over have to be charged with the correct amino acid or we're going to have problems. And I was going to say mutations in amino acyl tRNA synthetases, I was going to say that would cause a problem, but it would cause such a problem that they don't exist because an organism can't live unless the genetic code is acting generally correctly. I hope that makes sense. All right, so I promised you, I don't want to disappoint you, that we would talk about some general differences with respect to eukaryotes in this process. The logic of the process, you guys, is identical in eukaryotes and prokaryotes, in everything. By the logic, I mean just the general idea of how the information gets put from RNA language into amino acid language. But there are some significant differences. So now that we understand the process in bacteria, let's just observe what some of these different processes are in eukaryotes. Number one, the initiator tRNA, it may sound like a little picky thing, but it doesn't carry formal methionine. Let's draw a little sign like this. Is that how you do it? It carries normal, unformulated methionine. It's just the way it is. So if you see FMET, you know you're dealing with the bacterium. But methionine is always the first amino acid that comes over, okay? So in eukaryotes, it's just regular old methionine. Additionally, there is no equivalent to the Shine-Dalgarno site, or sequence, I should say. This was strange to begin with, but remember that eukaryotic RNAs are processed in a way that prokaryotic ones are not. So it seems clear that, remember that 5' cap that we discussed? That doesn't go on bacterial mRNAs. The cap interacts with the ribosome in a specific way. It binds, and then the mRNA gets scanned until the first AUG codon is found. So, you know, the AUG still has to end up in the right position, but it gets there by a different mechanism. And it seems to be because of the cap. Okay? Now, I don't think you're probably going to be required to know all of the details. I could be wrong. But ribosomes are larger in eukaryotes. They have a large subunit and a small subunit. Remember, there's one extra ribosomal RNA in eukaryotes as well. They look fairly similar, but they are larger. More proteins, more RNAs. It's not a big deal, but it's a fact. Now, This picture, you may say, whoa, and I'm almost saying, whoa, right now. There's a lot going on in this picture. But what we thought we would do is, at least in a eukaryote, put together all of the things that have to happen. Notice what I call this here, a summary of protein synthesis. I had told you not everyone does it this way, but many people use the term protein synthesis to mean the entire process from DNA to protein. So we've gone over it all, but this picture has everything in there. So let's see if we make sure we understand what's going on here. Now I'm just using the principles are similar for bacteria, but in a eukaryotic cell, there's more going on because you have RNA processing, you have a nucleus there, etc. So here's the DNA, right? Number one, DNA is in the nucleus, and they say that the first step is that transcription occurs. But remember, you get this primary or pre-mRNA that has introns and exons, and then RNA is processed before it leaves the nucleus. So remember, processing involves capping, tailing, and getting rid of the introns so that we have this mature RNA. The mature RNA leaves the nucleus, and I'm not positive if we said this. It's in my mind that we did, but in bacteria, transcription and translation are coupled. There's no nucleus to separate a messenger RNA that's being made from the ribosomes. They're all in the same compartment. Unlike that, in eukaryotes, the mRNA has to be fully completed, released into the cytoplasm before it can possibly think about interacting with the ribosome. So this looks like just one long piece, but it's meant to be a flowing sort of idea diagram. So the messenger RNA eventually gets out into the cytoplasm, and then pretty much what we just said starts happening, right? The message starts associating with ribosomes. You know, we went over all the details, but you have this initiation complex. Transfer RNAs are out there, they're charged, they're ready to go, and they're going to carry amino acids over to the ribosomes. And this here is a depiction of the process that we were just discussing, where tRNAs recognize and bind to mRNA codons, and this procession just continues. Bring a new one, transfer the peptide, translocate. Do it over and over and over and over again until you're done. So here you see a growing polypeptide coming out of there, a little bit longer than ours, but remember, real ones. I mean, some proteins are short, like 40 amino acids, but most of them are like 1,000. And then ultimately the polypeptide grows, and ultimately it's done, right? And that polypeptide will be released. So we have, at least the way I'm saying it, protein synthesis, including both transcription, translation, and all, you know, the steps in between, processing, etc. Everything that has to happen to get information that's encoded in the order of DNA bases ultimately to be encoded in a protein in the order of amino acids. Okay? So a lot going on in that picture, but it does basically give a flowing sort of feel to all of the processes that are necessary for expression of the genetic information in a protein. The ribosome structure is very well known, and while I doubt that anyone would ask you to draw one like this side view really shows you we do know exactly what's happening, and I think that the side view is particularly illuminating, and our artist did a great job, I think. Don't you think? Because look what's happening. Here's the small subunit down here. You see the messenger RNA is sort of threaded through there, right? But imagine the process that we just discussed going on, and each of these balls is a amino acid, and the polypeptide is exiting through this chute. That's not the same as the E site, because the E site has to do with transfer RNAs exiting. But as the polypeptide grows, and you can see it coming out, it's being threaded through there. I mean, this is a little machine. It's an amazing little machine. So I like this picture as though you can see, in a sense, that, you know, whatever's been read already is equivalent to the amino acids that are on that polypeptide. And this is going to keep growing, right, as we're reading more and more and more of the messenger RNA. The front view, you might say, is a little bit more boring. This is the way maybe you usually see it, but it shows you that, imagine that the ribosome is basically zipping down. It's moving along this transfer RNA as translocation occurs once after, again and again and again and again. And that's basically how we're going to read this thing. Now here you don't see the peptide coming out, but eventually you would. It would be coming out sort of through the middle up here from the front view. Okay? So the whole thing just fascinates me, and I hope it does you as well. One thing that I don't think I ever thought about too much before someone told me is that if you think about it, once a messenger RNA, we said this as a process, like start, go on, and stop, and that's true enough. But once a ribosome starts going and translating a messenger RNA, who's to say that they're long enough? Why can another ribosome come along and start translating too? And it turns out the answer is yes. And it doesn't always happen, but it's the rule rather than the exception. So I say here, in fact, more than one ribosome is usually translating the same message at the same time. And we look at that whole big structure and call it a polyribosome, or some people shorten that and just call it a polysome. But check this out. It's very interesting and it makes a lot of sense. So here's, again, it's a bit idealized, but we have a messenger RNA. There's the 5' end, there's the 3' end. So this ribosome here, it started down here, and it's been moving along, adding amino acids to the point where now we have this protein that's even starting to fold up, you can see here. And the point of this is, though, the same process is going on. But once this got out of the way, then another ribosome came. As soon as one gets out of the way enough, another one comes, another one comes, another one comes. Now, if this really is the end here, this one's just about to fall off and release that protein. But then this one will, then this one will, then this one will, then this one will, and more will be coming. So this is important for a variety of reasons, and this occurs in eukaryotes and prokaryotes, guys. You can even visualize it under the electron microscope. But it also is a good time to mention a process that's often referred to as amplification. And what that means is one messenger RNA might give rise to many, many, many copies of protein. It's not as if, at all, one messenger RNA encodes one type of protein, but it may be able to make 10, 20, 50 copies of that protein because it just keeps getting read over and over and over and over again. So one molecule has the information to make one type of molecule, but you can make a lot of it from that. That's really interesting for a variety of reasons. I mean, this has to do with gene regulation, so we can't really go there too much. But a viable question is, well, how much protein do you really want to make off of a messenger RNA? If it stayed around forever, in theory, you could just keep churning protein out, and that wouldn't generally be good. So one of the ways protein synthesis is regulated has to do with, this is a little bit higher level, but it has to do with the stability of the messenger RNA. In bacteria, very frequently, there are messenger RNAs whose half-life is like 5 minutes, and I'm not kidding. Because after that 5 minutes, they decay, and whatever protein has been made, that's enough. Because when things change, you can turn on another gene and start saying produce this protein, but you don't want the original one to keep getting made. I mean, situations are all different. But how long the messenger RNA sticks around is a big part of how much amplification is going to occur here. In humans, it's not uncommon to have a messenger RNA stick around like 24 hours, but the DNA sticks around your whole life. Hopefully it stays in good shape, right? RNAs are very, they're vital, but they're going out and being made all the time, and I hope it makes sense why that has to be the case. And the proteins are being made all the time and decaying as well. It's the only way we can exert a sort of dynamic control over the situation. Okay, so polyribosomes. Now, the last thing I wanted to discuss, I know this is a bit of a long video, but we had a whole discussion about mutations, but I want to talk about mutations here because we just understood protein synthesis. Now we want to understand both the terminology, and there are some problems that I suspect will come to you that are rather difficult that make use of some of this information. So mutations have been named based on what effect they have on protein synthesis, and in particular, what effect they have on the protein that a gene encodes. So number one, it may be obvious, but this is very important to note. I say, for example, a missense mutation is a change in a codon. It is a change in a codon, but remember this. Mutations always occur at the DNA level. That's where they're enshrined, if you will, because everything else sort of decays very quickly. So if a mutation occurs in the DNA, though, it's going to be copied and will end up in the RNA, right? So if there's a change in a codon so that a different amino acid is inserted, that is called missense. Now, some textbooks have a little variability to this. I say possibly that results in an altered phenotype because it does depend which amino acid is substituted for another. If it's very similar, nothing might happen. And put a little asterisk there because neutral mutations down here at the bottom of the page are an example of that. So some people, to make it clear, say that a missense mutation not only changes the amino acid, but it changes it in a way that does affect the phenotype. We talked about sickle cell anemia, for example, and the change in hemoglobin. That is a missense mutation. One amino acid is switched for another, and that leads to bad consequences, right, for the protein and for the individual. So that's absolutely a missense mutation. Let's skip nonsense just for a second so we can see what I'm trying to contrast with a neutral mutation. It is an amino acid change occurring, but there is no change in phenotype because the amino acid's similar enough, it doesn't change the protein enough that there's anything that can be detected that's different. So I don't want to overcomplicate this, but depending on how the terminology is used, you could make the case that a neutral mutation is sort of a type of missense mutation if all missense means is that one amino acid is switched to another, right? Because neutral is just clarifying that in that particular case, that change did not result in any phenotypic difference. Some people use missense to mean, yeah, the amino acid changed, but there is a phenotypic difference, and I wish that there was 100% clarity on that, but I don't see it. A nonsense mutation, on the other hand, means a definite thing, and that changes any codon that codes for an amino acid into a stop codon. What that does is cause premature termination and a truncated protein, because if all of a sudden it's going along and says, what? A stop codon is here. All right, I'm following instructions, I'm stopping. The protein, depending where this occurred, maybe it was supposed to be 1,000 amino acids, now it's only 400. It's not guaranteed, but it's very, very likely that that's going to cause a big problem in that protein. So a nonsense mutation is more likely to cause big problems than the other two. We have another one called a silent mutation. So in this case, it's a little bit of a subtle idea, but remember, mutations are occurring at the DNA level, right? So what if a base pair change does not cause a different amino acid to be substituted? This is because of the degeneracy of the code. It's because of what we discussed in the last session. CCC means proline, CCA means proline, right? So if a DNA mutation occurred that ended up changing the codon CCC to CCA, literally you would still make the exact same protein. And we said that we believe that's what degeneracy is for. I mean, it can't protect everything, but in those type of mutations, nothing changes. Literally, it's not even just being close. The exact same protein will be made, and that's why they call it silent. It's a mutation happened, but it literally, if you just looked at the protein, which is the expression of that information, you would never be able to know. Okay? We discussed when we were talking about how the genetic code was cracked, as it were, and how it was determined that it was a triplet code. We talked about frame shift mutations. So just to reiterate, what the effect would be on a protein. It's the addition or deletion of one or more bases, but what it really means is one or two. Three, remember, generally causes it not to be so bad, or multiples of three, whether they're added or deleted. So it usually results in a non-functional protein. I note here that multiples of three are less harmful. They may totally be unharmful. It depends. And that's why you can't make a massive generalization about frame shifts. But the whole idea of a frame shift is, since the code is non-overlapping, right? You read one codon, you read the next one, you read the next one. That's what it is. Any addition of one or two throws off the reading frame. And every codon, it says it right here, wherever that is, that addition or subtraction occurs, all the amino acids after that point are going to be altered because you're reading them in the wrong reading frame. It may result even in a different protein, a smaller one or a larger one, because when you're reading it that way, there could be a stop codon created, or the stop codon could be missed. So a lot of different things can happen with a frame shift. There are few guarantees in life, but more often than not, it does cause a large problem in the protein, often a very, very, very bad one. Okay, the last part of this, I wish we could stop and take a break now, but this is the perfect time to say it. Intergenic suppressor mutations. It's a mouthful, but when we were talking about mutations in general, remember we talked about suppressors. And in that context, we said, in life this doesn't usually work out, but in the case of a suppressor mutation, two wrongs somehow make a right. And now we're in a position to really understand at the molecular level how that could occur. So intergenic simply means not in the same gene, right? A mutation in another gene which sort of compensates for another mutation. So how could this occur? Well, these intergenic suppressors usually involve, again, it happens in the DNA, but it ends up changing a tRNA anticodon so that it now reads a mutation as normal. I hope that makes sense, but we're going to draw it out. So for example, let's say there was a nonsense mutation, right? Creates a stop codon. So that can be suppressed if the tRNA anticodon changes to be able to recognize that stop codon, right? So I'm going to write a little example of this. I'm going to draw it out on a clean page. Let's say you have a codon UAC. And UAC means tyrosine. I don't have that memorized, which is why I have to look. So that's the codon, right, in the messenger RNA. There's 3'. Let's say that changes to a UAG codon, which means stop, right? So that mutation is a nonsense mutation. It's going to cause a truncated, shorter protein, and that's the mutation, right? Now, normally, a tRNA would come over under the total normal circumstance, right? The anticodon of this tRNA would be AUG, and it would have tyrosine on the end of it, right? That's before any mutation took place. Once this mutation takes place, this tRNA is not going to come over there anymore, right? Because it's now a stop codon. That's why it's the mutation. But how would an intergenic suppressor mutation occur? What if this happened first, and then in the transfer RNA, we had a mutation where the anticodon changed from AUG to AUC. Now, wouldn't this be complementary? So mutation 1, which was in the messenger RNA codon, is compensated for by mutation 2, which is in the tRNA anticodon. Remember something important. Just because this anticodon changes, remember that aminoacyl tRNA synthetases don't care about the anticodon. They're still going to put tyrosine on this, and I've seen so many problems where this screws people up. Please remember that. Just because the anticodon changes, it does not mean that the amino acid changes, and in most cases it definitely remains the same. But can you see how this would suppress it? Because now when the alleged stop codon comes up, a mutated tRNA carrying tyrosine says, oh, I don't care. I mean, it doesn't have knowledge in the sense that we do. It binds to what's complementary to. So when it sees the stop codon, it says, oh yeah, I recognize that. And it brings the tyrosine over, and all is back to being well with the world, right? So that's how a mutation in a tRNA gene, for example, can suppress or compensate for a mutation in a messenger RNA codon. Just for the record, if you had a stop codon, right? I'm saying that was supposed to be there, all right? So this is a similar situation, but I'm saying here we had a situation where it was supposed to be tyrosine, but it mutated into a stop codon. And then the second site mutation compensated for that. What if you have something that's supposed to be a stop codon, stop, and you have this similar type of mutation that we just saw here? I just want to bring home the point because problems will be about this, guys. Practice them. If now a tRNA has AUC on it, when it was actually supposed to stop, now it's going to be inserting the amino acid tyrosine and continuing along its merry way. So if you have, I know this isn't a second site or suppressor mutation, but I'm just, as long as we're on the topic, if you have a mutation in the transfer RNA anticodon loop and it's not compensating for anything, if it happens to be able to recognize a stop codon, you're going to be getting a lot of longer proteins than you ever wanted, and that's a big problem. Okay? I hope that makes sense. So this is just sort of a summary of it. If a mutation occurs in the tRNA anticodon, I say either with or without an original mutation to suppress, it's going to recognize the normal stop codon, at least in our example. I mean, all sorts of types of things can happen that you would have to apply this type of thinking to. So as I just drew, it's going to recognize the normal stop codon, insert the amino acid, and continue going. If it was a suppressor mutation, things would go back to normal. If it was, as I just drew, it was supposed to stop, now it's going to keep going. You're going to get a longer protein because it won't stop until another stop codon, perhaps that's different than the one that tRNA can recognize, is encountered. But mutations in tRNA anticodons can have various consequences, I say, depending on the situation. But keep these principles in mind because when you get problems, a lot of times they'll be about a change in a codon, a change in an anticodon, how they go together. Is it a stop codon? Does it stop inappropriately? Does it continue inappropriately? I know it's a lot. But please remember this. I said it, but now here it is written in big bold print. Changing the anticodon loop does not change the amino acid attached to that tRNA because the enzyme that puts on that amino acid doesn't really care about the anticodon loop. Even though that seems odd, that's the fact of the matter. All right, well, that was quite a lot even for me. So this is so important, guys. I think you need to go and meditate about it for a little while or do something, or even take a break and then come back to it. Hopefully it all makes perfect sense for real. But if it doesn't, let it sink in a little, maybe watch again, and then I'll see you again real soon after you do that.